Maya Casey. And Maya will talk about, she also worked with Regan Herrera and Henk van der Ploeg, and she also had a mentor that was from the Sigler Consortium. Cody Sheik joined us from University of Minnesota, Duluth to work with Maya. So we thank Cody for that. And Maya, you are up.
Great. Can everyone see my screen?
Yes, it looks great. Thank you.
Okay, so hi everyone, thank you for that introduction, Mary. Yes, so I'm Maya, and I am super excited to talk to you about what I was doing this summer, which was exploring the buoyancy potential of Microcystis, which is a genus of cyanobacteria, using metagenomic data. So first, let's just talk about metagenomics more broadly. Metagenomic data refers to a sample that's taken directly from the environment that's then analyzed for all of the different genetic communities existing within that sample. So I like to think of it as essentially a snapshot of all of the different bacterial communities existing within a certain point. And just to give you an idea of how vast metagenomic data can be, on the right you can see a tree showing all of the different bacterial species we found within our data, which as you can see, there are quite a lot. So we know what type of data I was working with, but what exactly was I trying to get from that data? So my first research question was, do we see buoyancy-related genes or microcystin genes during the peak of a cyanobacterial harmful algal bloom? So we're interested in buoyancy and microcystins because these are two core components of Microcystis as a genus. Buoyancy is important because Microcystis can regulate its position in the water column depending on where optimal environmental conditions are. And then microcystins are the key toxins produced by Microcystis. So I hypothesized that we would see the key genes from both gene clusters present during the 2018 peak. But what do I mean by key genes? Well, at the bottom you can see the operon for MCY, which is corresponding to microcystin or toxin production. But an organism doesn't actually need all 10 of these genes in order to produce toxin. Instead, it only needs key genes, one of which is the MCYE gene. So in other words, when we see MCYE, we can say, okay, this is probably going to be a toxin-producing organism. My second research question is, what is the spatial distribution of the 2018 cyanobacterial hab in Western Lake Erie? So this is a satellite image showing the hab from 2018, and you can see that it's pretty spaced out. And so this question really aims at getting at which strains and species of Microcystis are we seeing at different points throughout the sampling locations. And so it's trying to see, do we see different abundances of strains and species at different locations? So my data came from the 3G ESP long-range underwater autonomous vehicle, which I'm sure as many of you know is at the forefront of HAB's tracking technology. The 3G can collect all sorts of types of data, but mine was primarily focused on just the genetic data. And my data came from the 2018 deployment in Western Lake Erie, which ranged from about late August to early September. And you can see the sampling sites on the left, and these will be shown a lot throughout this presentation. So the general workflow is that once that 3G sample made it back to the lab, DNA extraction was carried out, and then those samples were sent off for shotgun sequencing. Once those samples came back, a series of bioinformatics steps were carried out in order to limit the data to strictly microbial, since that's what we were primarily interested in. And so then this is where I came in. So all of that was done before my fellowship. And then when I came in, we basically took that data and assembled it by location. And so once we had those assemblies, we were able to do something called binning. And we used K-base, which is the primary genetic pipeline. So bins, which are also known as MAGs, which stands for Metagenome Assembled Genomes, are essentially that. You're organizing all of your data into different genomes. And this is really useful because once you have these genomes, you can conduct a series of different analyses. The first is taxonomy. So we used a taxonomy app in K-base, and basically this went through all of those genomes and just labeled them. So it said, okay, this genome's Microcystis, this genome's Pseudoanabana, this genome is non-cyanobacterial. And this is really helpful because it allows us to know exactly what we're looking at. So instead of seeing bin 1, bin 2, bin 3, now we're seeing, okay, Microcystis species or Anabana. And so then we can pick out the ones that we're actually interested in. The second analysis we ran was looking for the genes. And so we put those genomes in a gene annotation program. And then we were able to search for specific genes using BLAST. So I'm sure many of you have experience with BLAST. It's that huge database of all of the genetic data, but we actually just made our own little mini databases with just the genes that we were interested in. So the buoyancy genes and the microcystin genes. And then we were able to, remember at this point, we did have the taxonomy. So we were able to say, okay, this microcystin genome has these genes. And it allowed us to see, again, what we're working with. Finally, the last thing we did was we looked for relative abundance. And so remember that we're sampling at a lot of different sites. And so we're likely to have repeat genomes across those sites. And so first what we did is we got rid of those repeat genomes and picked one core genome for each species. And then we ran a program called CoverM, which goes back to our original data for each sample. And it says, okay, how much of this data in this sample maps back to each genome? And so then this tells us what percentage of the data is made up by each genome, hence the relative abundance. Okay, so getting into results, the first results I'll talk about is the occurrence of microcystin and buoyancy genes. So first talking about microcystin gene presence. So we picked out 6 bins that had these genes pop up, and you'll see that there's a varying level of specificity from strain to species. I'll explain the strains in a couple slides, but in terms of species, you can see that it's very generically named. And this is just because the taxonomy didn't actually give us the names of these species. So I just dubbed them A through D to differentiate them. And then you'll see at the bottom that there are the 10 different MCY genes, which I had shown earlier. And remember that we're looking for that MCYE gene as one of the key indicators of a toxic species. And so you'll see that almost all of these do have MCYE. What's really interesting is, for example, you'll see Microcystis species B only has MCYE and none of the others. And one explanation for this is the completeness of these genomes. And so you'll see that it has a completeness of about 32, which means that a lot of the data is still kind of floating around in our data set and just didn't get mapped back, which is okay. Seeing the MCYE is a promising indication that it's probably a toxic species. But again, it's a really low completeness, so that's probably why we're missing those genes. However, you'll note that for strain Y, for example, it has a pretty high completeness of about 75. And so we don't see any of the MCY genes for that strain. And so this allows us to pretty confidently say that this is a non-toxic strain. And so I've starred all of the strains that we would consider toxic, strains and species that we would consider toxic, and then I have strain Y, which we would say confidently is a non-toxic strain. Okay, so now talking about buoyancy genes. The gene cluster for buoyancy is the GBP gene cluster. And similar to MCY, we do also have key genes for GBP that we look for. So one of those is the GBP-A cluster, which is those first 3 on the left. And these are really important for the structure of gas vesicles. And so you'll note that the first 2 have most of the genes. We can probably say these are buoyant strain species. The second 2 are missing all of them, so we can say these are probably non-buoyant. But what's really interesting is those last 2 are missing the GBP-A genes, but happen to have a lot of the other GBP genes. And so this brings up a really important question of, are there other mechanisms or genes that we aren't seeing that maybe make up for those missing GBP genes? And this is beyond the scope of the data that we have, but it's definitely something that is interesting for future research. And so to summarize again, those first 2 are definitely buoyant, second 2 are probably non-buoyant, and last 2 I put an asterisk. The data would say probably not buoyant, but again, we can't say anything with confidence because we're not really sure what's going on there. And so to summarize, I took out the 4 genomes that had a completeness of 70 or higher. And so we can see we have a range of toxic and buoyant species, 1 toxic and non-buoyant species. And then I put that strain Y. We definitely know that it's non-toxic, but we don't know, again, if it's buoyant or non-buoyant. So I didn't want to confidently say anything because, again, we can't really tell from our data. Okay, so now the second results are the spatial analysis of Microcystis blooms. So if you'll remember, for the relative abundance, we took out all those extra genomes. And when we did this, we had 2 Microcystis genomes left. And what's really interesting is these were the exact same species, which is that I think it's 0022-82935 species. And so because it outputted these 2 species, we have to assume that they're different on the strain level. And so that's where strain X and strain Y come from. So we mapped the relative abundance. And so for strain X, you can see, just to orient you, this is showing a zoomed in version of the different sites in Western Lake Erie. And the size of the circle as well as the darkness of the circle corresponds to abundance. So generally the darker the circle as well as the bigger the circle, the higher the abundance. And you'll note that it only goes up to about 2%. And this is because, remember, we're looking at a metagenomic sample. So we have a lot of different species and we're only looking at Microcystis. So it's not going to make up too much of the sample. And so you'll see there's a pretty normal distribution at different sites have different abundances. And then for strain Y, we see a pretty similar distribution, but I would like to draw your attention to the key, which shows that the top threshold is 1.9% as opposed to 2%, which we saw with the previous strain. And while this might not seem like that big of a difference, when you put them side by side, there's actually some pretty big differences between the abundances of these two strains. And so on the left, you're looking at strain X, and on the right, you're looking at strain Y. And I'd like to point your attention to this particular point. So for WE16, for both of these strains, we saw the highest abundance. But for strain X, it was about 2.2, and for strain Y, it was about 1.7. So this is a pretty big difference between these two strains, and you'll also know other differences between abundances of these different strains. And so this is really interesting because what we're seeing is we're seeing the same exact species, but the abundances are different on the strain level. And so why are we seeing this? Well, if you'll remember, the strain X was a toxic species and strain Y was a non-toxic species. So one potential explanation is maybe there's something going on genetically that's affecting the abundance of these different species, strains. And so that's something that's really exciting, is that we're seeing such different abundances between different strains. And this definitely is a jumping off point for future research. Okay, so to conclude, we saw that different strains and species of Microcystis have a different assortment of GVP or MCY genes. So that's going back to that Venn diagram. We saw, toxic and buoyant species, toxic, non-buoyant species, and then again, that question of non-toxic, is it buoyant, is it not buoyant? But the point is we were only looking at about 4 of those Microcystis bins, and we're seeing such big differences between those. And so that's exciting because it's showing that there's a lot of different gene presence in different strains and species. The second big conclusion is that the abundance of genomes was dependent on both location and strain. And so we definitely did expect to see different genomes, different abundances of genomes at different locations. That's to be expected. It's kind of what Mia was talking about in her presentation as well, that there are so many factors that affect the abundance of these different genomes. But what's really interesting is also, again, that we saw different strain abundances. And this is something that's really exciting, and I'm excited to hear the future research that comes out of this. Okay, so there's a lot of future work that can come from the research that I did. First, I just want to put out there that my original project was supposed to compare the 2018 bloom to the 2019 bloom. And so if you'll note from these side-by-side pictures, the 2019 bloom was in much higher severity than the 2018 bloom. And so comparing a smaller bloom to a bigger bloom would help reveal a lot of really interesting questions about what's going on genetically. And also, it would be really interesting to see what strains and species pop up in the bigger bloom. And so this is a really cool and future project that I think will definitely be done, and I'm invested and excited to hear what we get from that. The other future direction is I only looked at Microcystis in my project, and this again is a metagenomic data that we're dealing with. And so we found a lot of different cyanobacterial species, as well as some pretty novel ones that when we looked them up, we couldn't find too much literature about them. And so this is exciting because looking more into the composition of these blooms and seeing the abundance of these other species that we maybe know less about will be really exciting and just teaching us more about what we're seeing in these blooms. And so with that, I would like to extend a huge thank you to Reagan, Cody, and Hank for answering my endless questions and being a constant support system this summer. And to the Sigler Fellowship Program, Mary, Michelle, Aubrey, and the other fellows. And I'd also like to extend a huge thank you to Paul and the 3G ESP team for collecting and organizing the data for this project. It made my job so much easier to have the data organized the way that it was. So thank you to everyone involved. And I am happy to take questions now.
Fantastic job, Maya. That was super interesting, and I loved the graphics on your slides. Okay, so if people have questions, you can, I'll leave it up to you if you'd like to enter them into the question box, or you may also raise your hand and I can unmute you. We've got plenty of time for questions, because Maya was right on time. Okay, we've got one from Anna Bagold. So Anna asks, what is the most interesting thing you learned during this project?
That is a great question. So I will forever be excited to talk about how when I started this summer, I didn't really know exactly what the steps we were doing meant. So you remember I showed all those different analyses we were doing, and then at the end to see it all come together. So I think one of the really cool things I learned was just about like trusting the process in bioinformatics and understanding that like everything has a role, and so that at the end you can kind of see the full picture. So I learned a lot of really cool bioinformatics skills, as well as just like how to work with big data sets, which is something that I am excited to take with me in the future.
So Anna just followed up to say that she thought the entire presentation was really interesting and opened up a lot of new research questions. So that's great feedback. Thank you, Anna. So I had a question, Maya. So you're talking about non-buoyant strains of Microcystis, and which made me wonder, and I don't know if you've discussed this with your mentors or thought about yourself, about what would be, are they at a competitive, are those strains at a competitive disadvantage because they aren't able to regulate their buoyancy in the water column? And what might that mean that we're seeing a mixture of buoyant and non-buoyant strains?
Yeah, so that is a great question. I just want to point out that, and I didn't mention this in my presentation, but the 3G was only sampling at one depth. And so future research would definitely need to look at other depths to see maybe we're missing like some of the buoyant strains. But I just want to also say that I think non-buoyant doesn't necessarily mean that it wouldn't be competitive, because it kind of depends on what's going on in the environment that you're looking at. And we kind of talked a little bit about this. I believe like we were talking about how like at one site we see like everything's like floating and there are a lot of buoyant species, but at another like completely different location, everything isn't floating, like there aren't buoyant species. And so seeing that like maybe there's something environmentally like more broadly going on that affects what we're seeing. So it's not necessarily maybe a competitive disadvantage or advantage, but rather it just has to do with like where they're found. But again, like this is something that's like exciting to look more into in the future.
Yeah, exactly. Thank you for those thoughts. It seems like, so Greg Dick, I think was asking a little more about that same topic. So Greg says, it's amazing you're able to get that far on genetic data during a summer project. Fully agree with that statement. He says, I wasn't aware that some Microcystis strains lack gas vesicle genes. Is there a precedent for that in the genomes of cultured Microcystis?
I do not know. I actually think that the GVP gene cluster needs to be continually studied, because I think that just understanding like the different GVP genes and like which ones actually do help with like gas vesicle formation is something that's still like not fully fleshed out, I guess. And so, yeah, I don't know about any precedent, but I think that this is something that is definitely needing to be continued to be studied.
Yeah, it sounds like it. So a little more feedback. Reagan says, great job, with 2 exclamation points. And Greg says, very interesting, thanks. So many follow-up questions. So Greg might be reaching out to you to follow up on some things, and with Reagan. So Maya, thank you so much. Fantastic job. It sounds like you've got a lot of us thinking. So that's fantastic.
Thank you.